Good day and thank you for attending this talk. The rules in assessing the marine systems go very deep in time, especially for freshwaters. Later in the 70s, Pearson and Rosenberg model started to help developing methods to assess the status in marine waters, and there are several. And was in 2000 when the AZTI Marine Biotic Index, the AMBI, was created at the same time as the Water Framework Directive in Europe, and this leads to a lot of publications related with different biotic indices based on the same approach. And even now we have the GAMBI, which is the genomic AMBI, so there are indices based on molecular methods. And also the same approach has been applied to other taxonomical groups like foraminifera and bacteria. What is the principle of this? Our idea was, if we are able to analyze the community and assign each species to an ecological group regarding the tolerance to the pollution or the pressures from sensitive to opportunistic, and then calculate the proportion of each group in the community, if there is a dominance of sensitive species, we have a good situation, a high situation in terms of quality. If there is a dominance of the opportunistic species, we have a worse situation, and in between we have the rest. When monitoring and assessing macroinvertebrate communities, we took the samples, we pick up all the individuals, identify them, count, and make the calculations of the biomass. But also when we have a lot of samples to analyze, this has high economic cost and long process, taxonomic expertise needed. So the alternative was doing an approach based on genomic. And in 2014, we published the GAMBI, which is the genomic AMBI. When we compare the taxonomic composition between the genomic and the morphological, the number of species matching is between 20 and 65%. But in 2018, we published this paper in which we compare the AMBI, morphological AMBI and GAMBI, and we can see that the number of reads fits quite well either with abundance or biomass. So instead of using presence absence, we can use the number of reads as biomass or as but the problem is that when we assess the status, we use MAMBI, which is the multivariate AMBI, which includes not only AMBI but also richness and diversity, and this was intercalibrated. So we need to compare the MAMBI, calculate with morphology, with MGAMBI, calculate with richness, diversity, and AMBI based on genomics. But we need to check the relationship between morphological and genomic results, especially the reference conditions for each type within the community. But for doing that, we need some standardization to make comparable morphological and genomic data. For example, we left only genus and species level, except in Nematoda and Nimirtia, which are used in AMBI calculations also. Then all species and genera under the Oligochaeta, Insecta, and Diptera were summed up. We removed all taxa with less than 10 reads by replicate. And then, as MGAMBI is a factor analysis, to assess the status, we need reference conditions, both of body status. In the case of GAMBI, richness and diversity based on molecular, we use the same bad conditions, which is associated conditions as in AMBI or MAMBI, sorry. But for the high status, we need reference conditions adapted. And for the different types we have in the Basque Country, which are 3 estuarine types and 2 coastal types, we calculate values for GAMBI, J richness and J diversity in order to do a better calculation of the MGAMBI. These are the results for the first 3 years. In the case of AMBI and GAMBI, the relations still is very good, as demonstrated in 2014. And when we look at the different types, it looks like this is okay again. When we look at richness, compare morphology and genomic, there is more or less good correlation, but there is not so good in terms of the step, because it should be like this. So this is because of the differences in the different typologies we have. And in the case of diversity, you can see that there is a dispersion in the comparison between morphology and genomics. And this comes from the dispersion within each of the types. When we calculate MGAMBI with the reference conditions for the different types, you can see that the correlation is not very good, and we can modify sometimes the reference conditions in order to get a better one, but still it is not as good as the comparison of AMBI and MGAMBI. The other approach we use for assessing the status is in bacteria, in which we developed the MicrogAMBI index, which was developed in 2017, and then in 2018 we tested it in different areas. The idea is the same. At the beginning we have a list of 226 taxa, and now it is more than 1,500 taxa, from which indicators of pollution were 39 at the beginning, and now they are 800 taxa. And instead of 5 groups, as in the AMBI, we use only 2 ecological groups. 1 sensitive to pollution inputs and 1 tolerant to pollution inputs. And using a more or less similar equation, we are able to calculate for MicrogAMBI the proportion of the sensitive and tolerant species, taxa, and taking into account the relative abundance, we can go to the high ecological status, good ecological status, moderate, depending on the proportion, poor and bad when there is a dominance of the ecological group 3. And with these boundaries, we calibrate and validate this, taking into account the sediment quality index, in which we included metals and organic compounds. And you can see that there is a good correlation between pressure index and MicrogAMBI with an asymptotic relation. As I commented you, we tested this in 2018, in different parts of the world, from the Arctic to the Antarctica and the Equator and the temperate areas. Just 2 examples, one in Korea, in which we tested this in a submarine entry of wastewater, which in high tide there is a good situation for bacteria, but in low tide and ebb tide we have a worse situation for the bacteria, and then again recovers. And when we compare the total nitrogen inputs in the area or concentration in the area with the MicrogAMBI, you can see that there is a clear correlation and good correlation, significant correlation. With low concentrations, the situation is high or good, and with high concentration of nitrogen, the situation is bad. In the Antarctica, we studied a gradient of eutrophication from eutrophic area to oligotrophic area, and you can see that in the eutrophic area the situation is bad. Then in this area it's moderate and good, or moderate good, and finally in the oligotrophic area the situation is high. This year we have published this paper in which we compare the 5 years pressures index average with the MicrogAMBI using a lot of data from the Basque Country, and there is a good correlation, but the cap analysis shows a bit mismatch between both. In the case of the comparison between a morphological AMBI and the MicrogAMBI with bacteria, so benthic communities and bacteria, the correlation is good, but still there is a small mismatch. And also we have published this paper on the application of MicrogAMBI in a continuum soil river lagoon or lake. As conclusions for macroinvertebrates, we think that we have successful results with GAMBI with immediate application, but we have problems when using mGAMBI. For example, differences in diversity and richness compared to morphological results. Setting adequate reference conditions for molecular methods is needed. We need to have the whole gradient of degradation for each type. The intercalibration is needed for the application in the Water Framework Directive, and we expect still 3 more years for adequate comparability. For bacteria, in the case of MicrogAMBI, we can assess the status of bacteria in a cost-effective way. The current list of taxa requires a scientific consensus to make it useful everywhere. The boundaries between quality classes should be adjusted. And finally, there are still shortcomings in using genomic methods, for example, in standardization and comparability, but they can be overcome in few years. In our opinion, the future of monitoring and assessment could be based mainly in genomic methods if we solve the above problems. Thank you very much for attending this talk, and this presentation is available in my ResearchGate profile and in YouTube.